this cytorial, we're going to talk a little bit about fluidic systems in flow cytometry and understand how different flow cytometers manipulate cells and sheath fluid. Let's remember that there are some fundamental aspects of flow cytometers. Cells are always in suspension. The usual fluid for cells is saline. The sheath can be saline or water, and the sheath must be saline for sorting. Next, we want the cells to pass in a single file through the illuminated volume of the laser beam, and the cells will eventually pass through a very narrow orifice between 50 and 300 microns. There are three basic fluidic systems in flow cytometers: positive pressure systems, peristaltic pump-based systems, and syringe-based systems. Positive pressure systems have been around for years, and gas pressure drives the sample through the flow chamber as well as using sample pressure. And it uses the well-known principle of hydrodynamic focusing in the flow chamber. More about that later. When we change the pressure of the sheath, we change the flow rate of the sample. Then there's a technology using peristaltic pumps that delivers sample to the flow chamber. It's a slightly different delivery method, and it's much easier to manage from a hardware perspective. Let's look at a typical instrument that uses a peristaltic pump. Here's a peristaltic pump with the sample collection point here. The peristaltic pump is often placed quite close to the flow chamber to reduce the distance of sample travel. And finally, there are syringe-based systems. In a syringe-based system, we have a calibrated syringe that pushes a fixed sample through the tubing to the flow chamber. These are called highly quantitative systems that can perform absolute counts because the exact volume delivered is known. In these systems, the sample is pulled into the sample line from the sample tube and then pushed back into the flow chamber. In this way, it's possible to measure the particle number in the fixed volume of sample, and we call this absolute counting. Now we know how the sample is physically delivered to the flow chamber. What happens in the flow chamber itself? While I often draw the flow chamber facing down, which is how it works for sorters, analyzers usually face it upward. We have a special tube for the sample delivery, and we have inlets for sheath fluid. The laser beam passes through one part of the chamber, which, as you note, it narrows, and Howard Shapiro calls this the neck-down region. It's important to note, because of the hydrodynamic focusing, the sample sheath gets squished a bit and becomes a very narrow core, maybe 20 microns or so. Because of this narrowing of the sample stream, the velocity of the cells increases and they start to really fly through the chamber. The whole idea behind hydrodynamic focusing is to make sure that the cells are in a single file. This way, we can hope to measure only one cell at a time. Of course, the goal is to establish what we call laminar flow. I guess I better remind you about hydrodynamic focusing. The sheath flow principle was developed by a scientist by the name of P.J. Crossland-Taylor in 1953. What Crossland-Taylor determined was that you could create laminar flow for your cells if you injected the cells into the center of a faster flowing stream. This aligned the cells into a narrow core within the wider stream. So the principles of fluid dynamics creates a sample within a sample. If the two fluids differ enough in their velocity or density, they do not mix. They form a stable two-layer flow, which reminds me whether or not the flow will be laminar can be determined from Reynolds number. It's not as complicated as it might look. It basically says that laminar flow, which means stable flow to all extents and purposes, is a function of the diameter of the tubing, the density and the viscosity of the fluid, and the fluid's velocity. Reynolds determined that when Re was less than 2300, which by the way is a dimensionless number, that flow is always laminar. But when it exceeds 2300, flow is likely to be turbulent, something we do not want to have in our flow cytometer. In case you want to see some different flow chambers, I have a few photos. In some instruments, you get a bunch of different flow chambers, mostly sorters, but in analyzers, the flow chamber is usually fixed. The picture in the center is pretty interesting. This is the chamber in the cytoflex, and you can see the sample delivery tube at the bottom with some green dye running through the system. And behind that is where the signals are collected by the fiber optics. On the right is a sorter chamber that I sliced down the middle so you can see what is actually inside. Now, a minute ago I mentioned that in all analyzers, the flow path is facing upward. The reason is that sometimes you get bubbles in your sample, and bubbles tend to go up so they don't stick around causing turbulence in the flow chamber. Now, a small note about sample pressure and the sample core. Below we have two identical flow chambers, but the one on the left has low sample pressure and the one on the right has higher sample pressure. What you should note here is that when you increase the sample pressure, you will widen the sample core. The risk is that some cells may not be in the same vertical plane and that you might impact the quality of your signals. It's something to keep in mind when you push those cells faster through your flow cytometer. Talking about samples and pressure, some instruments have large containers of sheath and waste, so they might operate continuously for a very long time. Others are much smaller. It's important to remember that the sheath and waste tanks are always the same size. When the waste fills up, there's a shut-off to stop the instrument till the waste is emptied and the sheath tank is refilled. Well, I've tried to give you a better idea of how fluidics in flow cytometry works. There are many more important issues, but I think we covered the basics. There is one more thing I want to remind you about. Every time you collect data, don't forget to collect one more parameter: time. It's important that you always collect sample number versus time on every sample, as this gives you confidence that you're getting a constant flow rate during the collection period. Many folks forget to collect these parameters, and you should make sure you're not one of them. It's a critical QC principle that was established by Jim Watson back in the 1980s. If you don't remember who Jim is, go look up his work and take a look at this manuscript. So whenever you're making a histogram of a dataset, make sure you also collect time so that you're sure your sample rate is stable. Okay, that's it for this tutorial. Thanks for watching.